Bismillahirrahmanirrahim. Now I'm going to discuss magnesium and manganese metabolism. Before going in details of their biochemical functions and biomedical importance, I must tell you the difference between the two type of enzymes, which are called as metal activated enzyme and metallo enzymes. Because these metal ions and trace elements, either they are the component, they are belong to the class of metallo enzymes, or they are being required by the metal activated enzyme. So one should have a clear concept what is a metal activated enzyme and what is a metallo enzyme. Metal activated enzymes actually show great catalytic activity by the presence of a mono or divalent metal ion exterior to their protein. Means the metal ion is not the part of the enzyme protein. They are required there in the assay medium, but they are not the integral part of the structure of enzyme protein. And metal activity enzyme typically lose activity during purification. Example of the metal activated enzyme is hexokinase, which require magnesium. Kinases is a class of enzyme which belongs to transferases, and they transfer phosphate from ATP to the substrate. So here hexokinases means K phosphate is being transferred either to the glucose, to fructose, or to the mannose. So it is metal activated enzyme. Metallo enzymes are those enzymes where the metal as a cofactor bound covalently to their structure. Either the metal is present at the active site of the enzyme or at the substrate binding site of the enzyme, and that metal cannot be separated out because it's covalently attached to the enzyme protein. So until and unless the enzyme protein is broken down, it will not be separated out from the enzyme protein. Example is cytochrome oxidase, which require heme in the form of Fe3 plus and the this copper as a cofactor. And then catalase again requires a heme, and you are very well aware heme is a combination of this protoporphyrin 9 and the ferrous. Now this slide shows you the magnesium. One of the substrate is kinases, as I told you earlier. They transfer phosphate, and always the donor of phosphate is ATP. So there is a structure of ATP. You can see nitrogenous base adenine, pentose sugar ribose, and three phosphates are part of the ATP, adenosine triphosphate. And you are very well aware that phosphate are negatively charged molecules. Now magnesium is a divalent cation. It has a two positive valency. It is present in the form of two positive valency, and here it is serving two functions. Either it is forming a chelate with the substrate. A chelate is the combination of a metal line with the organic substance, or it is neutralizing the negative charge of the phosphate. Phosphate is negatively charged and magnesium has got a positive charge. So here this metal cofactor magnesium is always required by kinases, and it's either form metal chelate formation, or it neutralizes the negative charge which is present on the phosphate of the ATP molecule. Now, one important thing regarding magnesium, that it is the fourth most abundant cation in the body. We have sodium, we have potassium, we have magnesium, we have manganese. So it is the fourth most abundant and the second most abundant in intracellular cations. It is mainly seen in the intracellular fluid. The total body content of the magnesium is 25 grams. Out of this 25 grams, 60% is present in the bones, and there it complex with the calcium and phosphate. 30% is present in the soft tissues, and only 1% is present in the extracellular. Now this picture shows you the balance between the intake and the output. You can see here our daily requirement is 12 millimoles per day. Out of this 12 millimoles, 6 millimoles are being absorbed from the GIT, 8 millimoles are being excreted down via feces. Those 6 millimoles then goes to the plasma in the extracellular fluid, and daily you can see 84 millimoles of this extracellular fluid when the extracellular fluid plasma is filtered through glomeruli, 84 millimoles of the magnesium is filtered. Out of this 84, 80 millimoles is reabsorbed. Only 4 millimoles are excreted in urine. So this is very interesting that 12 millimoles is a per day requirement. 8 millimoles are lost in feces and 4 millimoles are lost in urine. So intake is equals to the output. The rest goes to the bone, as we have seen in the last slide, that 60% is present in bones along with the calcium and the phosphate, and 30% of the magnesium is present in the soft tissues. As far as the dietary source of magnesium is concerned, cereals, beans, vegetables, potatoes, meat, milk, fruits, fish, they all are the ideal sources for the magnesium. And mainly it is absorbed from the small intestine, as we have discussed earlier, that out of 12 millimoles, 6 millimoles is absorbed, rest is excreted out in feces. And there are certain minerals are there or trace elements are there which competes with the absorption. And calcium, phosphate and alcohol decreases magnesium absorption, whereas the parathyroid hormone increases magnesium absorption. The normal plasma magnesium concentration is between 1.7 to 3 milligrams per deci. Now, there are two major roles of magnesium in the biological system. Number one, that it can compete with the calcium for binding sites on proteins and membranes. And second is that it can form chelates with important intracellular anionic ligands like ATP. I told you earlier, chelate is a combination of metal ions with the organic molecules. So either it competes with the calcium or it form chelate. Now magnesium acts as a essential core factors for a number of enzymes, and mainly those enzymes which are related to the cellular respiration, that is the oxidative phosphorylation, the enzymes of the glycolytic pathway, and the transmembrane transport of other cations such as calcium and sodium. The activity of sodium, potassium, ATPase depends on magnesium. Now remember one thing, where there is a ATP, magnesium will be requirement of that enzyme or that process. So sodium, potassium, ATPase, they are the pumps which are present in the cell membrane, and they are responsible for the transport of sodium, potassium with the requirement of energy as ATP. The permeability characteristics and electrical properties of the membranes are affected by magnesium. Reduced extracellular magnesium concentration, they increase membrane excitability in tissues such as heart. Magnesium also acts to maintain low resting concentration of intracellular calcium ions. So this is a very important thing one must remember, that magnesium maintains low intracellular calcium ions. So when there is a magnesium deficiency, more and more calcium ion is there in the intracellularly, and extracellular calcium ions are decreased. So and it competes with the membrane. We have just discussed earlier that it competes with calcium for membrane binding site, and it stimulates the separation of calcium or movement of calcium from the sarcoplasmic reticulum. This is a list of the functions which are being served by the magnesium, or magnesium is required for these biochemical functions. Number one is formation of bones and teeth, which we already knew that 60% goes to the bones. And then it is required to maintain neuromuscular irritability. It is important in the maintenance of membrane electrical potential, and this role is linked to the potassium and the calcium. It is also required for skeletal development, and it is required for the maintenance of electrical potential in nerve and muscle membrane. more than 300 enzymes require magnesium as a cofactor. Again, I will repeat, cofactor is a metal ion which is covalently attached to an enzyme, like hexokinases, glucokinases, phosphofructokinases, pyruvate carboxylase, peptidases, ribonucleases, and adenylate cyclase. So you can see very important enzymes: hexokinase, glucokinase, phosphofructokinase, and pyruvate carboxylase. These four enzymes are mainly concerned with the carbohydrate metabolism and with the glycolysis and gluconeogenesis. Peptidases are mainly concerned with the breakdown of the protein. Ribonucleases has a role in nucleotide metabolism, and adenylate cyclase has a role in the mediation of hormone action. Now, this is a very important function of the magnesium. As we in the previous slide, I told you that magnesium maintains low intracellular calcium concentration. So, more calcium is extracellularly when the magnesium is in the normal concentration. So, you can see here in a healthy cell where there is a proper magnesium level is magnesium in normal concentration, the intracellular calcium ions are less. The C indicates calcium and M indicates the magnesium. So more, as we discussed earlier, it is the second most abundant intracellular cation, magnesium itself, and it keeps intracellular calcium concentration at a low level. But what happens in unhealthy cell, or that cell which is not healthy, what happens, and there is a magnesium deficiency, so less magnesium ions are intracellularly and more calcium is there. So hypomagnesemia means mia means blood and hypo means less. Hypomagnesemia is always associated with low serum calcium concentration, because now the calcium will be intracellularly and extracellularly calcium concentration will be less. So low levels of magnesiums associated with the low levels of calcium in the extracellular fluid, and more calcium inside the cell. Now here we see the effect of magnesium on individual organs and systems. As far as the cardiovascular system is concerned, it has a blood pressure lowering effect, and it also reduces the heart rhythm problems. In muscles, it counteracts with the calcium, as we discussed earlier, that it competes with the calcium for binding on membranes for binding sites, and it inhibits the over excitability of the muscles, and that's why it reduces cramps. As far as the central nervous system is concerned, it also reduces the excitability of the nerve cell and has a calming effect. And it is essential for energy metabolism, for ATP synthesis. When we say energy metabolism, it means we are talking about the ATP and the metabolism of the lipids and the carbohydrates. And I told you earlier that hexokinase, phosphofructokinase, glucokinase, pyruvate carboxylase, they all are enzymes related to carbohydrate metabolism. And it is also important for protein synthesis. Now, as with like other minerals, where we discuss the extremes of the whether they are when consumed in excessive amount or whether there is a deficiency. So hypomagnesemia means, mia means blood, hypo means decrease. So decrease magnesium in the blood. And in one slide I told you about the normal levels is from 1.7 to 3 milligrams per deciliter. So if the serum magnesium level is less than 1.7 milligram, we will declare or label that individual as suffering from hypomagnesemia. Now, what could be the possible reasons for this decrease in magnesium in blood? Number one is maybe there is a decrease intake due to the malnutrition, or there is a decreased absorption due to the malabsorption, or it may be lost in diarrhea and vomiting, or there may be causes which are related to the kidney, because we know that 4 millimoles are being excreted in urine via kidney. So due to renal tubular acidosis or as a result of diuretic therapy. Now, renal tubular acidosis. In renal tubular acidosis, what happens that magnesium is being lost in urine because it carrying the positive charge, or when a diuretic therapy is being given to the individual to treat hypertension. Now, clinical significance. As a result of these causes, when the magnesium level is less than 1.7 milligram per dl, the patient will present with certain signs and symptoms, and the clinical manifestation of the magnesium deficiency is characterized by impairment of neuromuscular junction. We are very well aware that it is required for electrical potential, maintenance of the electrical potential and the membrane potential. So there is a impairment of neuromuscular function. There will be hyperirritability, and remember, magnesium is low, then serum calcium concentration is also low, and both has a role in that in causation of the sign and symptoms. Hyperirritability, tetany is because of the hypocalcemia, convulsions both because of the magnesium and the calcium, and the changes on the ECG will also be observed. Now, epidemiological evidence relates low magnesium intake to the high incidence of cardiac death. If there is a low magnesium, it may lead to cardiac, individual may die of the heart attacks. Hypertension, myocardial infarction, cardiac dysarrhythmias, coronary vasospasm, and premature atherosclerosis also have been linked to the magnesium depletion. We discussed hypomagnesemia when the values are less than 1.7 milligrams per dl. But if the serum magnesium concentration is above 3, because the upper limit was 3, 1.7 to 3 milligrams per dl is the normal serum magnesium concentration. So when the concentration is more than 3.5 milligram per dl, we will call it as hypermagnesemia. Causes may be, though this hypermagnesemia is very rarely observed, but it is usually seen in patients who are suffering from the renal failure, and as a result of this, due to renal failure, the excretion of magnesium via urine or via kidneys is decreased, so there is a accumulation of magnesium in the circulation. Or when there is a excessive intake, either orally or parenterally. When we say parenterally, means by via injection, intramuscular or IV, or when there's a endocrine disorder that is called as hyperparathyroidism, when the parathyroid hormone is being released in excessive amount. In hypermagnesemia, there will be depression of the neuromuscular system. If you recall, in hypomagnesemia, there was excitability, irritability, tetany, convulsions. Here there is a depression of the neuromuscular system. There will be lethargy, hypotension. Hypermagnesemia is characterized by hypotension, and hypomagnesemia is characterized by hypertension. And here the pulse rate will be, heart rate will be decreased, that is the bradycardia. Now I will discuss the second trace element of the today's topic, and that is the manganese. And it is a transitional metal found in the biological system. Manganese is a transitional metal, means those metals which have whose valencies can be changed. So it is present either in a 2 plus or 3 plus valency state, and before that we have discussed those transitional elements that includes iron, copper, and now it is manganese, and it is mostly present in the circulation bound to a protein. The functions which are mainly concerned with the manganese are that it is concerned with the formation of connective tissue and bony tissues. So here one thing is very much clear, that just before we have discussed that beside calcium, phosphate, magnesium, manganese is also present in bones, and it is a component of the connective tissue. Manganese is required for growth and reproductive functions, And as far as the carbohydrate metabolism is concerned, it is a requirement for a process that is called as gluconeogenesis. Gluconeogenesis is a process by which the glucose is synthesized from non-carbohydrate sources like amino acids, like glycerol, like or fatty acids, and it is also a requirement for the lipid metabolism. As far as the dietary sources of manganese are concerned, manganese-rich sources includes whole grain foods, nuts, leafy vegetables, soy products, and teas. A median intake in the United States is about 2 milligrams per day. So, approximate intake per day is 2 milligrams. And as far as the absorption, transport, and excretion of the manganese is concerned, it is absorbed from the intestine either by active transport or facilitated diffusion. So much is not known about its absorption, whether it is a facilitated diffusion or it is an active transport. So active transport is that transport that requires energy in the form of ATP. And so far, no specific manganese transporter proteins are identified in the GIT. Evidences have indicated that several iron transporters are involved in manganese absorption. So, if you recall, beside transferrin has a role in the transport of iron, and it also is required for the transport of selenium. Selenium also required transferrin for its transport via circulation, and here we are coming again and another trace element or mineral which require iron transporters for its transport into the circulation. Once it is absorbed, manganese is transported in the portal blood to the liver bound to albumin. After absorption, it is in the circulation, it is bound to the albumin until it reaches to the liver. So it is exported to other tissues bound to the transferrin and possibly by a Q macroglobulins. Excretion of the manganese is via bile into the feces, and its urinary output is very low. So major route of excretion of manganese is via feces or GI. Now, in the beginning of this lecture, I have given you a description of metallo enzymes and metal activated enzyme. Magnesium was metal activated was belong to the class of the metal activated enzyme, but manganese is a constituent of many important metalloenzymes, and it is also a non-specific enzyme activator. A number of enzymes require manganese. Among these, we have superoxide dismutase, pyruvate carboxylase, arginase, and glucosyl transferase. Superoxide dismutase, a very important enzyme which has a role in the detoxification of superoxide radical, a type of free oxygen radical which is being synthesized continuously as a result of oxygen toxicity and as a result of the normal metabolism. And in the topic of zinc and copper, we have discussed that zinc, copper, and manganese, they are the three trace elements which are required by this enzyme superoxide dismutase. And this superoxide dismutase converts superoxide radicals into the hydrogen peroxide, and then the catalase and peroxidases, they convert those hydrogen peroxide to the water molecules. So we have been gone through this reaction a number of times. Then pyruvate carboxylase. Pyruvate carboxylase is a important enzyme of a carbohydrate metabolism that is called as gluconeogenesis. So gluconeogenesis is a process by which, or a metabolic pathway by which non-carbohydrate substrates are utilized to produce or to synthesize glucose. Then one more enzyme, that is arginase. Arginase is an important enzyme of a pathway that is called as urea cycle. And then glycosyl transferases. Glycosyl transferases are actually responsible for the transfer of glucose residues to the proteins like proteoglycans or to the glycoproteins. And you are very well aware, just before we have discussed that manganese has a role in the formation of connective tissue and bones. Now, connective tissues or the ground substance or cementing substance, they are made up of structural proteins like collagen, elastin. Beside that, they require carbohydrates in the form of heteropolysaccharides or in the form of GAGs, and also proteoglycans. So for the formation of proteoglycans, we require an enzyme that is called as glycosyl transferases. So here, this is a very important thing which you must remember. As far as the connective tissue formation is concerned, the components of the connective tissue, I will again repeat for your convenience. It is structural proteins like collagen, elastin, carbohydrates in the form of heteropolysaccharides or glycosaminoglycans, and we proteoglycans. So proteoglycans are the protein when conjugate with the carbohydrates. Uh, um, more pro more proteins and less carbohydrates. Okay. And also these are it is required for cartilage formation. So it has a role. Manganese has a role in the structural integrity of the bone and skin, and it is also important in context of the normal wound healing. As far as the recommended daily allowance of the manganese is concerned, the Food and the Nutrition Board has set an intake of around 2.3 milligrams per day for males, and they have recommended one intake of 1.8 milligrams per day for females. Some inherited manganese transporter defects are being identified, and they have an important role in the differential diagnosis of the pediatric movement disorders. So manganese has a role, manganese transporter defects being related to the movement disorders in children. Pediatrics, when we say that, it is related to the children. So systemic manganese accumulation leads to distinct syndromes of hypermagnesemia, characterized by when the manganese levels are being increased, because when the transporters are not there, it is being there accumulated within the body, and it will lead to increased levels of magnesium in the blood and manganese level in the blood, and it will be characterized by polycythemia, dystonia, chronic liver disease, and depletion of iron stores. Now, these movement disorders in children can be diagnosed on two bases. Number one, on the basis of the MRI brain, and the blood manganese levels will be drastically increased, rather 10 times than that of the normal. So highly raised levels of manganese and MRI brain will help to confirm the diagnosis of movement disorders in children.